right, number 15. Explain how restriction enzymes and ligase can be used to insert a piece of DNA into a plasmid. All right, well, yeah, for the plasmid, as far as it's concerned, right, the restriction enzymes are going to cut our plasmid usually at two different sites. And what this does now is it produces our plasmid with this cut site here where we can insert a gene. Now, usually we use restriction enzymes that produce sticky ends, and I'll talk about this more in the next problem, why we do that. But regardless, we've cut the plasmid now so that we can potentially insert a gene into here. Now, as far as the piece of DNA that we want to insert, now there are several different approaches we can use here. Right? One is we can potentially just cut the DNA straight out of the genomic DNA and insert it in here. Right? So in this sort of strategy, we have our whole genome. Here's our gene. And maybe we make a cut site here with a restriction enzyme, and then also here with a restriction enzyme. And this whole piece here contains our gene. And we can basically mix that cut piece of DNA with our cut plasmid and add in DNA ligase. DNA ligase is an enzyme that will join together cut pieces of DNA to make an intact piece of DNA. All right, so one option is we can do something like this, basically cut our gene out of the genomic DNA. Now, what we're actually going to be doing in lab is we are going to be amplifying your gene with PCR. So we will have just your gene of interest and a lot of it because we'll have amplified it. Now on the ends of the gene, we're going to add on restriction sites. These two restriction sites, one at the front of the gene, one at the back of the gene. All right, so when we now digest this product with our restriction enzymes, we're going to get a gene fragment that is now complementary and can be inserted into this cut site of the plasmid and then ligated into place.